Welcome to the WTR Small Cap Spotlight podcast. I'm your host, Tim Gerdeman, Vice Chair and Co-founder and Chief Marketing Officer of Water Tower Research. In today's podcast episode, I'm being joined by Dr. Lior Shaltiel, CEO of NurExone Biologic, along with Yoram Drucker, the company's founder, VP Business Development, and active chairman. Also joining is my Water Tower Research colleague, Robert Sassoon. NurExone Biologic is a biopharma company focused on developing regenerative exosome-based therapies for central nervous system injuries. Its lead product, XOP10, has demonstrated strong preclinical data supporting clinical potential in treating acute spinal cord and optic nerve injury, both multi-billion-dollar markets. NurExone Biologic is listed on the OTCQB under ticker symbol NRXBF and on the TSX Venture Exchange under ticker symbol NRX. So without further ado, let me welcome the gentleman to today's call.
Hello, good morning.
Hi, good morning.
Great to be here.
Lior and Yoram, let me kick off the podcast by asking each of you to tell our listeners about your professional backgrounds, please.
Okay, I will start with myself as the CEO of the company. I have a background in engineering and science. I have a chemical engineering bachelor degree, physiology as a master, and a PhD in pharmacology from the University of Munich, LMU. I returned to Israel in 2014, directly to the industry, working with different drug delivery system companies. And in April 2021, I joined NurExone as the CEO of the company. In parallel, I have also academic position at the Hebrew University Business School as the academic head of the biomedical program.
Thank you. Good morning. I'm a serial entrepreneur from Israel. I'm initiating companies more than 25 years. I was a co-founder of companies like Pluristem, Brainstorm. Most of my activities in the cell and gene therapy. Some of the companies that I initiated are public traded companies. And I'm also serving as a chairman and VP Business Development of NurExone, which I founded in 2020.
Great backgrounds. Thanks for that introduction, gents, and I'll turn it over to Robert.
Thanks, Tim. So there may be listeners to this podcast that are not as familiar with NurExone. So can you give us an overview of your development pipeline and the principal indications you're looking to address?
Sure. So the company came out as a spin-off from two elite university, the Tel Aviv University with Professor Dani Ofen, and the Technion, which is the MIT of Israel, with Professor Shulamit Levenberg. They both investigate technology how to regenerate nerves, especially in the CNS, central nervous system. The product of it, which is XOP10, this is the company lead molecule that we license from both institute, showing a very strong efficacy in regeneration of neurons, while our first model is in spinal cord injury, acute spinal cord injury. We have several animal model with up to 100% success getting them to move again.
And I would like to say a few words about the commercial side. As we're moving forward with the preparation for the clinical trials, we established a fully owned subsidiary in the US to serve as the production facility for our naive exosome, which are loaded with our specific molecule to treat the different indication in the central nervous system. And this production facility is expecting to start generate revenues second half of next year, 2026, as there is a huge demand for different indications of a naive exosome beside of what we are doing in our drug delivery platform in the Technion Haifa.
Thanks for that. So let's delve into your exotherapy platform and your lead candidate XOP10. First, can you actually explain what exosomes are and their function in the human body?
Sure. So exosomes are tiny particles that secrete from cells. They are expressed in any organism, like human, other animals in the in the biological system. And the idea is to communicate between cell to cell, between healthy tissue to a damaged tissue. And when we have an injury, like in spinal cord injury, exosomes are accumulate in the area of the injury where there is inflammation. And the idea is to, first of all, reduce the inflammation, and second, promote the regeneration. This is the activity. We try to help nature to do its job even better by adding siRNA into the exosomes and then promoting even further the regenerative property of those exosomes and creating true regeneration and neuron protection in order to get the signal across after the injury and show a functional recovery.
Thanks for that explanation. So now let's go on to discuss what XOP10 actually does. What is its mechanism of action and its role in the regenerative process? And further to that point, if we talk about your spinal cord injury, which is one of the indications that you're trying to address, why is there currently no function restoring therapy?
Yeah, so let's start with the product because I start to answer it when I explain what are exosomes. We need to understand that this XOP10, it's a combined therapy between the exosome property that has a therapeutic effect and the siRNA against the protein P10 that we loaded in to the exosomes. So the exosomes, as I said, are reducing the inflammation when they accumulate in the area of the inflamed tissue after the injury. The exosomes are engulfed into the cells, specifically to the neurons, and then promote regeneration with their own cargo. Addition to that, we have the siRNA that target specifically P10, which is a key factor, key protein in our cells. This protein keeps the steady state of the cells, not to proliferate, to stop any way of regeneration, and is very active in the CNS, not like in the peripheral nerves. So what we do is inhibiting the inhibitor. We try to reduce the inhibitor and then promote regeneration. And this is what we see in a tissue level and also in the animal scale and the animal level by seeing active functional recovery, which can be motor, sensoric, urine bladder, and even quality of life.
Seems remarkable that one therapeutic candidate can show such effectiveness across several severe and previously irreversible injuries. So
I will say more than that. I will say more than that, that we see this effect not only with the spinal cord injury, we saw it also in the optic nerve damage. We saw it also in the facial nerve. And we do believe that the same mechanism of action will be relevant for other indications such as traumatic brain injury. Those indications are even much larger than the spinal cord injury.
Right.
So the market is huge.
So potential for a significant expansion of indications. That sounds very promising. So what would you say differentiates your approach to other therapeutic methods targeting the same indications?
So I will answer that. There are four main differentiated points that I would like to mention. First of all, we are not a cell-based therapy. Most of the therapies that are now dealing with recovering spinal cord injuries or central nerve injuries are based on cells that are being proliferate, scaffold, growth, whatever, and they need to be growth, and they need to be implemented in the patient. So they have to be, must be, the same patient cells, because if not, you are creating a immune reaction, and those cells are going to be rejected. We are cell-free therapy. We don't need to do any kind of adaptation to the patients. We can do it as an off-the-shelf product, ready to be used, and that's it. Our exosomes are nanocytes, meaning that they can penetrate the brain, crossing the blood-brain barrier without any specific treatment or other molecules involved in the drug itself. We are administering our drug in a minimally invasive way, meaning that we don't need to touch the injury area. We don't need to implement anything. We don't need to go through any kind of surgery. For example, in acute spinal cord injury, our plan is to give a simple injection to the spinal cord fluid. The exosomes goes directly like a targeted missiles to the injury area and making their magic. That's it. We don't need to do any kind of surgery to optic nerve damage and so on. It's a big difference in sense of implementing it, the drug, from the physicians itself. And the last point, it's off-the-shelf product. You can use it immediately. For example, it might be that in traumatic brain injury, you will need to do an immediate intervention to secure as much neurons in the brain. So you would like to use a ready to be used off-the-shelf product, and we can offer it because the expected safety profile is very, very high. And I think those 4 points are making a huge difference between all the developments that are being done today to what we are offering to the market.
That sounds really interesting. And obviously, people will be interested to know how long this is going to take, to what your product roadmap is in your journey to commercialization.
So I will add to Yoram and also answer your questions, because this is the main change between other exosomes therapy that are working in the field. If in the past, most of the company would modify the cell source, okay, in order to produce specific exosomes that will include some kind of protein, which will be relevant. In our case, the modification of the exosomes is done post-production. So we have first produced our drug substance, which is the naive exosomes, and then load it with the siRNA. So it starts to be like a Lego. You can have the one block of exosomes and add different compound depending on your target. So the opportunities here are huge. Second, we are dealing what is the most important things in exosomes and in cell therapy is what is your source of cells, okay? And we can use one source of cells. In this case, we secure our raw material, our cells, which is fully GMP characterized, approved FDA cell, in order to produce all our variety of product, including, of course, XOP10, and also producing naive exosomes, as Yoram said before, for the cosmetic aesthetic treatment. So this is first. And when we are dealing with this commercially and regulatory, that's actually make our life much easier, because we don't need to communicate so much with the agency, FDA or EMA, to explain what is the source of the cell, because the cells are already, we have a number. The FDA, the agency, knows what is the cells of cells. They know that it's characterized, so we don't need to prove anything else. We need to prove the characterization of our exosomes.
Right.
When I'm saying that, this means that by next year, our plan is to get even to first in human, even before phase 1, and then I would say between 12 to 18 months to get the IND submission and official phase 1/2A for spinal cord injury, and can be also another phase 1/2A for the optic nerve damage, which is more in the direction of glaucoma, acute glaucoma, and other ophthalmological indications.
Right. So we're talking about milestones during the course of 26 and 27.
Yes.
Right.
Yes.
So finally, from me, maybe Yoram was speaking about this. Can you speak about your balance sheet and cash burn situation and your financial strategy to advance these programs?
Yeah, of course. So since inception, the company raised about $20 million US. We are supported by our investors and shareholders down the road. And as we are moving forward, we are raising by private investments, PIPEs, funds to the company. We announced a few months ago that we have intention, and we are working on uplisting to the national market in the US because we think that this is the place where the company should be, taking in account all the development and the things that the company is doing. And beside of the ways that the company is doing, the company is establishing Exotop, the US fully owned subsidiary, which we expect to start generate substantial amount of money end of 2026. So it will give us also some kind of secure funds and reduce future dilution to the shareholders. The company has sufficient funds to move forward. We are now in the process of accelerated warrants. We expect to get substantial amount of money from those warrants. By the way, in the history, we have more than 85% of warrants exercise in 2 rounds that were done already. Again, our shareholder community is very supportive, and they are believing what we are doing, and they are supporting the company as we're moving forward.
Thank you for that. So thank you, Lior and Yoram, for giving us a better understanding of NurExone and its novel therapeutic approach to what are already unmet medical needs. Definitely will be interested in monitoring your progress. And I'm just going to hand it back to Tim.
Thanks, Robert. Thank you for joining us today, Lior and Yoram of NuroExone Biologic, OTCQB, ticker symbol NRXBF, and TSX Venture Exchange, ticker symbol NRX. And thanks to Robert Sassoon for also joining us. Finally, a special thanks to the editor and producer of the program, Krista Fitzpatrick. Thank you for listening, and don't forget to subscribe, as well as visiting www.watertowerresearch.com to stay up to speed on the company's small cap written research reports, podcasts, fireside chats, industry-specific symposiums, and conference schedules. We will see you next time for another edition of the WTR Small Cap Spotlight Podcast.